welcome everybody, and it's good to see we have another good audience today. So today I'm going to talk about planning validation studies. So we saw yesterday when we were looking at the downloads of the Eurachem guides that validation was one of the really interesting topics for people. So I'm just going to talk about one particular aspect here, which is to do with how we plan an effective validation study. So I'm going to just briefly introduce the importance of planning, look at the content of a validation plan, look at some possible experimental designs, and then highlight some Eurachem guidance that's available. So why is planning so important? Well, I guess that any of you that have been involved with method validation know that it's potentially quite a complex and time-consuming activity, and it can generate a significant amount of data. So there are a lot of questions and decisions that we need to make when we start on a validation exercise. So which performance characteristics are important? What materials are we going to analyse? How many replicates are needed? How should the data be processed? And how are we going to assess fitness for purpose at the end of the study? So to ensure the validation study itself is fit for purpose, we really need to consider all of these issues before starting work. I think if we don't plan things properly, we might get there eventually, but it will possibly take us a bit longer and maybe not be as efficient as we would have hoped. And what I'm going to talk through is really the idea of using a planning template. And I'll show you later that there is a Eurachem guide that contains a suggested template. And if we use a template, it allows a more consistent approach. And also, if we start with a nice planning document, it can quite easily be converted to a validation report at the end of the process. So I'm just going to have a look at what might go into a validation plan. So we need to start by being clear about what it is we're validating and why we're carrying out the particular study. So really the validation should always start with a documented measurement procedure. I think sometimes there can be a bit of a blurred boundary between when method development stops and validation starts. But I think really we should try and draw a definite line and say, now I've got a method which is documented, and I want to take that forward for the formal validation. So this is where we're really setting the scene for what it is we're validating and why we're doing it. So obviously we should know what the method is, have any reference values, and it's probably useful to capture the status of this particular method. So is it a standard method that we're using unmodified in the lab? Is it a standard method that we've had to modify in some way? Or perhaps it's a method that's been developed wholly in-house. And then there might be different reasons for carrying out a study. And obviously that will relate to the status of the method. So is it a full validation of a new in-house method? Or maybe it's more of a verification type study where we're using a standard method, or perhaps we've made some changes and we just want to check that the method is still fit for purpose. So just to look at that in a little bit more detail, there are a number of reasons why we might want to undertake a study. So as I said, it could be a complete detailed validation of a brand new method, maybe checking that a published method, we can actually get it to work in our labs and that we can achieve any stated performance characteristics. It might be a validation as a result of change of scope of a method, or perhaps revalidation following change in operating conditions. And also potentially if a method hasn't been used for a while, we may need to revalidate after a period of non-use. Linked in obviously to defining the method that we're going to validate is having a clear statement of the analytical requirement. So what do we actually need our method to be able to achieve? And there's a number of issues we need to address here. So obviously what it is we're actually trying to measure. So what's the analyte? We need to be very specific there as to what it is we will actually be determining in our particular test samples. We should also have a clear statement of the measurand, so that's the quantity that we're intending to measure. So it's not just enough to state what the analyte might be. So for example, is it copper? We need to know, perhaps, is it the total amount of copper that's present, or is it the amount that's extracted under specified conditions? We should be specifying the measurement units that we're going to use to report our results, and also the required range. So what's the expected range of values that we're going to encounter in our samples? And then also we need to know as much as possible about the materials we're actually going to be analysing. So what is the matrix? What's the physical form that we might be faced with? Also need to understand what's the purpose of the measurement. So what are the results actually going to be used for? Because that will impact on our performance criteria. So in some cases, we may need a very high performing method that delivers results with a very small uncertainty, but in other cases, a larger uncertainty might be tolerable. So we should not only understand what it is we're measuring and what we're measuring it in, but also what those results will be used for. And obviously there's a whole range of reasons why laboratories make measurements. Could be to check compliance with a regulation or some kind of specification. It could be part of monitoring a production process, or potentially it might be part of directing a research and development project. I think when people think about method validation, probably what they're mainly focusing on is the performance characteristics of the particular method, and thinking about things like precision, bias, capability of detection. So clearly one of the key aspects of actually planning the method validation study is going to be deciding which of these performance characteristics we need to evaluate. And there's guidance in the Eurachem Fitness for Purpose guide as to how you might decide which of these performance characteristics you need to evaluate, because it's not necessarily the case that you need to evaluate every performance characteristic on every occasion. So this table is actually taken from the Eurachem Fitness for Purpose guide, and it's actually based on a table, I think, from one of the pharmaceutical standards for method validation. So that's why it's talking about things like impurities and main components, but the principle would hold across all different types of measurement sectors. Sometimes we're just interested in perhaps identification and not quantification. So in that case, perhaps a fairly limited number of performance characteristics might be of interest. But perhaps if we're looking at very low levels of an analyte, then we would obviously be interested in capability of detection. But if it's something that we're expecting to be there as a main component, then perhaps the capability of detection is not so important to us. But generally, we find that characteristics such as precision and bias and the working range and linearity of the method are pretty much applicable across a range of different validation studies and different requirements. So we need to decide, obviously, which performance characteristics we're going to study. But then we obviously need to decide how we're going to evaluate each of them. So this would really form the core of our method validation plan. So the template that's in the Eurachem supplements basically has a page for all of the performance characteristics, and you can see the information that would be required. So obviously which characteristic it is you're actually going to be determining. Might be useful to include a description of what that particular parameter actually tells us. What does it represent? We should ideally specify the performance criteria. So I'll say a bit more about that in a minute. We need to know how we're going to judge the performance of the method. So if we measure precision, how do we decide if that's actually acceptable or fit for purpose? Obviously, one of the really key components is actually specifying the experiments that we're going to do. So this will be the heart of our validation study, is actually generating the data that tells us how the method does actually perform. So we need to think about what materials would we be analysing. So do we need a certified reference material, for example, or maybe it's a performance characteristic that we could evaluate by analysing surplus test materials. One of the questions that always comes up when planning these studies is, how many measurements do I need to make? So what would be a suitable level of replication to allow us to get sufficient information to have confidence in our evaluation of that particular characteristic? And also, in what order are we going to do these measurements? So in a minute, I'm just going to highlight some possible experimental designs that we might use, but it is really important to plan the whole study, because sometimes we can find that actually we could get information about a number of different performance characteristics, but from the same set of data. We should always be thinking about data analysis at the planning stage. It's obviously relatively easy in the laboratory to generate lots of numbers, but then we have to decide what to do with them. And the statistical analysis shouldn't be an afterthought. It should be planned in to the study. We need to make sure we're clear on what statistical tools we might use to evaluate our data. So is it going to be something perhaps fairly straightforward, such as just calculating a mean or a standard deviation, or perhaps we're planning to use maybe some slightly more sophisticated tools, such as analysis of variance. So we need to make sure that we do end up with data sets that we can actually analyse and that would give us sufficient information to be able to get or to be able to draw reasonable conclusions from the data. So the statistical analysis shouldn't be a salvage operation that's applied after you've generated all of the data. It should be built into that planning, so we're clear how we're going to evaluate the data, but also clear on how we're going to assess fitness for purpose. So perhaps there might be some other statistical tests that we plan to do to allow us to judge whether the method is actually meeting the required performance criteria. And then the template just has some other spaces if there are any particular notes about particular performance characteristic, and perhaps an overall conclusion. So once the data have been generated, there should be a statement as to whether that particular performance characteristic has actually met the specified performance criteria. So thinking a bit more about the experimental planning, we need to think, as I said, not only what we're going to analyse in terms of the materials, but how we're going to do that. So how many measurements and in what order might we do it. So this is a key step, and what we're aiming to do is to maximise the information that we can obtain from any given experiment. So as I mentioned a moment ago, it might be possible to obtain information on more than one performance characteristic, but from the same set of experiments. There are obviously lots of different experimental designs available, and I've just picked out 4 here that are perhaps fairly common in validation studies, and these are the 4 that are mentioned in the Eurachem guidance. I'm just going to quickly illustrate 3 of them and say what they look like and what they might be used for. So I'm sure everybody has carried out a simple replication design at some point. That's something we probably do on an almost daily basis, where we just make repeated measurements on a single material, and we can calculate the standard deviation of those results to give us an estimate of the variability or the spread of the data. So this is a fairly common approach in validation for precision studies in particular, and especially if we're trying to evaluate repeatability. So just as a reminder, repeatability is when we're just making measurements over a short time frame in the same lab by the same analyst using the same equipment. So this kind of study lends itself very well to repeatability estimation, because we can just make our measurements within the analytical run and calculate the standard deviation to give us our estimate of the repeatability precision. If we were actually analysing a certified reference material as part of a simple replication study, then that would give us a reference value. So that would mean that not only could we get an estimate of precision from that data, if we have the reference value, we could also evaluate bias. So obviously we don't need a CRM to evaluate precision because we don't need the reference value. But if we were using a CRM, we could evaluate both precision and bias from that data. Another fairly common design that might be used in a validation study is what's referred to as a nested design. And this is where we have groups of results that are nested below a particular effect. And in validation, again, where that's quite useful is for precision studies, and particularly if we're interested in not only repeatability, but also in intermediate precision, if it was an in-house study. If it was a interlaboratory comparison for validation, then this is the design that's commonly used, but the replicates are grouped under different laboratories. For intermediate precision, the replicates are nested under the different analytical runs, and as part of the validation planning, we would need to decide what we're going to change from one run to the next. Is it just measurements perhaps made on different days, or potentially we could have measurements made by different analysts and possibly using different pieces of equipment. So when we have a design like this, it can be processed using one-way analysis of variance. So that's just an overview of the ANOVA table. But if we carry out analysis of variance, that generates mean square values, and from those we can calculate standard deviations. So if this had been set up so that these replicates were under repeatability conditions, then our S within that we could calculate would actually give us our estimate of repeatability, but we can also calculate the additional between groups, so between run variability as well. And if we were to combine both of those, that would give us an estimate of our intermediate precision, assuming this was a within lab study. And the Eurachem Fitness for Purpose guide actually gives us an example of how you do these calculations in one of the appendices to that guide. The other one I'm going to highlight is the fractional factorial design. So a factorial design allows us to study multiple parameters at 2 or more levels. If we were trying to carry out a full factorial, that would involve experiments using all combinations of factors and levels. And obviously that can be quite time consuming because the number of experiments required would multiply up quite quickly. So it's quite common to use fractional factorial designs, and that's where we have starting with a full factorial, but a carefully chosen combination of levels have been removed. So what I'm showing here is what's sometimes referred to as a Plackett-Berman design. So that is just a form of fractional factorial design, but it's one of the designs that's quite commonly used for doing formal ruggedness studies. So this design allows you to study up to 7 parameters. So we've just labeled them here A to G at 2 different levels. And you can do that across 8 experiments. And the grid is just telling you the particular combination of conditions you need for each particular experiment. So you're changing the factors from one level to another in an orderly way as defined by the experimental design. So obviously I don't have time to go into these in detail during this presentation, but it gives you an idea of what might be available. So next on the list was performance target. So we've looked at how we might decide which performance characteristics to study, and we've looked at some potential experimental designs to allow us to evaluate them. But obviously the final step of a validation exercise is to assess whether the method is actually fit for purpose. And I know from experience of running training courses on method validation that this is often one of the more challenging aspects for laboratories. How do you decide what is actually good enough? Where do you get the information from? Now, there is Eurachem guidance related to this on setting target uncertainties, so that is obviously a related issue, but for a lot of laboratories they're still needing to make a judgment on fitness for purpose based around specific performance characteristics. So where might you get that information from? So where could you get these target values? Sometimes you might be lucky, I guess, and it might actually be defined in a particular standard or regulation. So there are some regulations where laboratories are allowed to use a method of their choice, but they have to demonstrate that it does meet particular performance criteria. It might be specified by the customer. Perhaps this is not so common, but it might be that perhaps you have a customer who's had measurements being made over a period of time. They have a good understanding of what is required. Maybe they had a previous lab doing the analysis for them, but they know what performance was achievable there. So they might have some requirements in terms of what the method can actually achieve. It might be stated in a standard published method. So if you're using a published method, perhaps either unmodified, or maybe even if you have made some modifications to it, that gives you an indication of what should be achievable. So it's quite common to have criteria specified, particularly around precision, possibly capability of detection. So again, that gives you something to try to judge your performance against. Sometimes we might have to try and base the performance on similar procedures that are known to be fit for purpose. So it might be that perhaps one of the reasons for doing a validation exercise is that you're trying to replace one method with another, and you need to demonstrate that the performance of the new method is at least as good as the method that's being replaced. And finally, I think sometimes it can be that laboratories, particularly in a research setting, are really working kind of at the leading edge of what is capable in terms of measurement. So sometimes it might be that actually the purpose for doing this study is to actually define the current state of the art. So what is this type of measurement procedure actually capable of achieving? And then the final steps of this process should be an overall approval and some kind of summary of the method validation. So I realise now I'm crossing the line from planning into actually reporting, but it's obviously important that any validation plan should be approved by an appropriate staff member before the validation commences. But after the study is complete, the lab needs to produce a validation report. And as I mentioned earlier, using that template can assist with that, because obviously once you've got the data, you can include a summary of that within the template, and also the judgment of whether the method is fit for purpose. But it's also useful to provide an overall summary of the validation. So which performance characteristics were studied and how did they actually perform, and the whole thing should be completed by a final statement on whether the aims of the study have been met and whether the method is in fact fit for purpose. And then obviously there should be a final sign off of that validation report. So how can Eurachem help with this? Well, I've mentioned a couple of documents as I've been giving this presentation. So the main guide is the fitness for purpose of analytical methods. As you saw yesterday, as I said, that's probably our most popular guide in terms of downloads. It's currently under revision, but you can still access the second edition from the Eurachem website. And then in 2019, we published a supplement specifically on the planning and reporting of validation studies. So if you'd like more detail on the issues that I've been talking about this morning, then I recommend you have a look at that supplement. So just to sum up, validation should always be a planned and documented activity. For each performance characteristic we decide to study, we need to specify the materials we're going to analyse, the number of measurements, the order, the data analysis, and ideally the performance criteria. I've recommended using a planning template for a consistent approach and also for perhaps easy generation of a validation report at the end. And both the plan and the final report should be signed off. So thank you for your attention. I'm not sure if we've got much time left, but if there are any questions, I'll be pleased to try to answer them. Thank you.